Always the dominant gene will suppress the expression of the recessive gene. So Gregor Mendel, he is called as the father of genetics. What is F1 generation? F1 means, F means filial. That is the first form generation, filial generation. Test cross means crossing between the F1 hybrid with that of the recessive parent. Hello everyone, a warm welcome to today's session on Principle of Inheritance and Variations. I'm Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in this particular session, we shall deal about Mendelian inheritances of gene. So what are the different laws Mendel had proposed in order to study the genes? So we all know that in an organism, when it gives birth, the organism will look similar to its parent. Let's take the example of an elephant. So an elephant always gives birth to an elephant, right? Will it give birth to a deer or will it give birth to an earthworm or a parrot? No, the baby will look exactly like the parent elephant. Even in the case of human beings, always the child of a human being will have similar characters like that of the parent, and it will look a bit similar to the parents. It will not be like a human will give birth to an elephant or a deer or something else, right? Just like that, the mango tree, the seeds of the mango tree, when you sow it, it will always germinate and give rise to a mango tree. If you sow the apple seeds will germinate and give rise to an apple tree. It is not like the mango tree. From the mango tree, you can get a lemon tree or any other tree, plant, different species. So have you ever wondered why is it so? It is actually because something which was earlier called as factors were carried from the parent to the progeny. That is why they look similar to each other, or they carry at least one or two trait or character from the parent. So in this particular chapter, we will be discussing about how they are carried and why it is so, what it was later called as. So all these we'll discuss in today's session. Before moving to this session, it is our duty to pay a tribute to important scientists in the field of genetics, that is Gregor Mendel and Reginald Punnett. So Gregor Mendel, he is called as the father of genetics. So he is the father of genetics. So Gregor Mendel, he is called as the father of genetics because he was the first person to say that the progeny or the offspring inherit few of the characters or traits from the parent. So what he did was, he had a very beautiful pea garden, that is green peas, the pea garden in his house. And when he was looking at the pea plants, he saw certain variations in the generation of the plant, that is the first generation, the second generation. He saw certain variations, or he saw that the same character or trait was carried from the first generation, from the parent plant to the progeny, that is the first generation. So why all this happened? It was a question mark for him. So that is when he started to study, why is it so? Why does the progeny look similar to the parent? And that is the time when he discovered that some of the traits were carried from the parent to the progeny or the offspring. So how he did that, all that we will study. And also the credit for using the binomial expression or mathematical expressions or statistical methods to calculate or to find out the genetic variations, all that, or inheritance of genes, all that credit, it goes to Gregor Mendel. And while remembering Gregor Mendel, it is also important for us to remember Reginald Punnett. So you might wonder what Punnett has to do in genetics. He was the one to give something called as Punnett square. It is called Punnett square. So Punnett square is nothing but a box square wherein we put the alleles or the genes, so that we can cross and we can find out like how the progeny is going to look like phenotypically and what genotype or the gene is carried to the progeny. So it is a simple box-like structure which is used to study. So here we will write the male and the female character. Then if it is the genes, we will write here, or the gametes, we will write here. So this is how it is done. So this is nothing but the Punnett square. So we shall, or you will understand clearly when we are dealing with the coming slides. To give this particular square box to calculate like what genes will be carried to the progeny, that credit goes to Reginald Punnett. So these two scientists are very, very important. So next, moving on to Mendels Laws of Inheritance. So in Mendels Laws of Inheritance, he proposed two theories. So what are those two theories? To understand the theories, we have to first study the experiments that he conducted. So in the next session, we will study what are the two laws he has proposed. But in this particular session, we will only concentrate on what different experiments he conducted and what type of plants he selected, pea plants he selected for conducting the experiment. So Mendel, he performed various hybridization experiments. So I had told you, Mendel in his garden, he had pea plants. That is, he had a garden wherein lots of pea plants was there, and he conducted experiments using those pea plants to find out why the progeny looks similar to that of the parent. Why is it so? So to do that, he conducted various hybridization experiments. So if you look at my previous sessions, you know that in reproduction of flowering plants, that is sexual reproduction of flowering plants, there was a term called as artificial hybridization, which is nothing but cross-pollination, wherein cross-pollination is brought about in a plant in order to get different traits in the progeny. So that is the hybridization experiments that was conducted by Mendel on the garden pea plant, that is Pisum sativum. So what Mendel did was, for his experiments, he selected different garden pea plants, or he selected garden pea plants having different traits or characters in them, and those he considered them to be the parent. So what character the parent pea plants had was, first is he selected the height of the stem, wherein he saw that one of the parent plant was tall and the other parent plant was dwarf. And he also selected those pea plants with the different color of flower. He selected one of the parent plant with violet color flower and one more parent plant with white color flower. And also you should remember these two terms called as dominant and recessive. So we will look into that later. So apart from that, he also selected the pea plant in which he considered the position of the flower. So what he saw was, he selected a parent plant wherein the position or where the flower is situated, that is either it was situated in the axile, or in one more pea plant, parent pea plant, it was situated in the terminal. So axile means in somewhere in between the plant or in the internode region of the plant, the flower will be situated. Terminal means only at the tip the flower will be situated. So all these different traits he selected. Next, he also gave importance to the shape of the pod, that is the outer covering of the pea that is there, that is nothing but the pod. So this is the pod, and inside the pea will be present like this, seed. Pod and seed. So this complete thing that is there, this thing, it is the pod, that is the outer covering. So these things are the seeds inside, that is being present. So he selected the pod, shape of the pod. So one he selected that was inflated like a balloon. It was swollen, it was inflated like a balloon, and the other was constricted. So you might have bought pea from the market. Just observe the pods. Some will be bulged, the pods will be bulged. Some will be flat and inflated. So it is because of the different traits that have been carried from the parent. Based on that, you get the pods. So that is why. So next, he also gave importance to the color of the pod, wherein he selected the parent plant having green color pod and the parent plant having yellow color pod. And later he also gave importance to the shape of the seed. So he selected parent plants wherein one parent plant had round seed and the other one produced wrinkle seed, or in other terms, one parent plant, it produced pea seeds which was completely smooth and round, and the other parent plant produced pea seeds, though it was round, it was wrinkled or it was shriveled. Like if you keep the pea seed for a longer time, it will, like, wrinkles will start appearing on that, right? So those kinds he selected. And next he selected for color of the seed. So based on the color, he selected if the seed was yellow color or green color. So what he did was, based on these characters, few of them we will explain and take it by taking them as example. So he selected tall and dwarf and all that to cross-pollinate. So he cross-pollinated between the tall plant and the dwarf plant to check what the progeny might look like, whether the entire plant will be tall, whether few of the plants will be tall, whether few of the progeny will be dwarf, or the entire progeny or the offspring will be dwarf. So I told you about two terms called as dominant and recessive. So this is very important in terms of genetics. So dominant means here it will be T. So in T, I am taking the example of tall. So tall will have two alleles. So alleles are nothing but the genes that are complementing each other. So what are the two alleles here? T. It is the two alleles. So T, capital T, it is for dominant, tall. Being tall is always dominant. Being dwarf is always a recessive character. So here T, it is recessive. So say for example here violet will be having dominant gene. So WW. White will be having recessive gene, WW. Like that, he selected the dominant gene and the recessive gene. So this is about dominance and recessive. And always the dominant gene will suppress the expression of the recessive gene. Usually the recessive gene will not get expressed because the dominant gene will suppress the expression of the recessive gene. So this is about dominant and recessive. So next we'll move on to how Mendel brought about hybridization in order to bring about cross-pollination so that he can study those progeny, how will they look like. So what Mendel did was, he followed various steps in order to bring about cross-pollination in a pea or to cross a pea. So first what he did was, he selected two pea plants with contrasting characters. So if you select, say for example, if Mendel had selected both the parent plant which were tall, then no need of doing the crossing experiments, because even though the crossing takes place, it is understood that the progeny will always be tall. So that is why, to avoid confusion, Mendel, he selected two pea plants with contrasting characters. That is, one plant which was tall and the other one was dwarf. So to achieve cross-pollination and to prevent self-pollination, hybridization technique should be followed, wherein one of the flowers should be emasculated. So what is emasculation? The removal of the male reproductive part, that is the anther, from the flower. So what Mendel did was, in his garden, he selected two parent plants wherein one was tall and the other was dwarf. But here I have taken the example of the color of the flower. So we will consider the same. So what Mendel did was, he selected one pea plant, that is parent pea plant, which produced violet color flowers, and he selected one more parent pea plant that produced white color flowers. So always the anthers will be removed from the flower, that is, or the plant that is carrying the dominant gene. So remember what I told you, here the dominant gene is carried by the violet color flowers. So what was done was, according to emasculation technique, these anthers will be removed. The anthers will be removed. So since the anthers are removed, only the gynoecium is being present. That is why this will be considered as the female flower. It is considered as the female flower. And in white that is the recessive parent, in other plant, all the anthers will be intact. Why here anther will not be removed? Because here also, if the anther is removed in this particular flower also, then where will pollination occur if the pollen grains are not at all there, right? So that is why in the recessive parent, it will be intact. The anthers will remain intact only in one of the parent, that is in this case in the parent having violet color flower, the anthers will be removed. So this will be the female flower. So you all know that the female, to represent a female, we use symbol this one, and to represent a male, the symbol this is used. So it is easy to remember. Female women are always interested in the mirror, right? So it looks like a mirror, as if a mirror with a stand, she is holding the mirror. So that is why it is easy to remember. Male is considered to be dominant in a society. How much ever progress women have made, but still male is considered to be dominant. So that is why it is a progressive arrow mark like this. So female is always represented in this form and male in this one. So this is how it is represented. So what he did was he brought about cross-pollination. Now cross-pollination took place wherein the anther which produced the pollen grains, it fertilized the female flower and the ovule was fertilized the ovule and the seeds were developed. Now we have the seeds. So these are the seeds that were developed. So here you know this is the pod and the seed. So seeds were developed. So now these seeds, they were sown to the soil. So they were sown to the soil and Mendel waited for the plants to grow. So when the plants grew later on, he found out that all the plants had purple color flowers. All the plants, they produced purple color flower. Why? Because the dominant gene here was purple or violet or purple color flowers. Therefore, all the dominant character got expressed here. So this in detail we'll study in the coming session. So this was his conclusion. That is, something is carried from the parent. So now this is the F1 generation. So F1 generation. So I want you to know about F1 generation. So what is F1 generation? F1 means, F means filial. That is the first form generation. Filial generation, first form. That is F1 generation. So those plants, those seeds that you got by crossing the two different characters of pea plant, those seeds you have sown and the plantlets or progenies have come up. So these progenies are called as the F1 generation. Now if you make a cross between these pea plants and sow the seeds and you get again, you get the next set of plants, then it will be the F2 generation. Again, those seeds if you collect after cross pollination and sow it, it will be the F3 generation. Like this, the generations will go on continuing. So this is what Mendel did. So what he did was, first he selected two pea plants with contrasting characters. So here he selected especially one pea plant with the violet color flower and the other with the white color flower. He removed the anther, which is called as emasculation. So that, why is emasculation done? In order to prevent self-pollination and to promote cross-pollination. That is, if self-pollination occurs, then different characters will not get. If in the purple flower the anthers are not removed, then self-pollination will occur. Then the resulting plant, it is understood that it will become purple color only. Or if in white color self-pollination occurs, then the resulting plants will be white in color. So that is why, in order to avoid the confusion, emasculation is being done, so that to avoid self-pollination. So this is now the female parent, as I told you, because the anther is removed, it is the female parent. So that will be crossed with the male parent. It will be crossed with the male parent and the seeds are collected and the offsprings are produced. This is the steps that he followed. So any experiment Mendel conducted hereafter was according to this procedure itself. So we will study what all different kinds of experiments he conducted to prove this. So first what he did was, he went in for finding out what happens if a monohybrid cross is done. Mono, hybrid, mono means one. So here only one character or trait or one particular gene he took into consideration. That is one gene or one character or one trait from the male parent and one gene or one character or one trait from the female parent. So he did a cross or brought about cross pollination between two parents. What are the two parents? It is the parent which is tall and the parent that is dwarf. Parent that is tall and one parent is dwarf. So what happens here is when a cross pollination is done between these two, the resulting progenies, that is the F1 generation, will have what set of plants or what characters the plants will have, we'll just see. So what he did was, Mendel, he crossed a tall and a dwarf pea plant to study the inheritance of one gene. So what one gene he considered here now? The gene responsible for the height of the plant. So this plant has only one gene which is responsible for the tallness of the plant, and the other plant has one gene that is responsible for the dwarfness of the plant. So this is the parental plants. So these two are the parental plants. So he selected two parent plants, one which was tall and one which was dwarf. Then he did the crossing and he found out that in this particular monohybrid, that is mono, one particular gene or one particular trait, the phenotypic ratio and the genotypic ratio. What is phenotypic? Phenotype means phenotype. So it is the external morphology of a plant. External, or in other terms you can just say it is the morphology of the plant. So morphology means when you look at a plant, you can say whether the plant is tall or whether the plant is short, whether the plant produces, whether the color is white or whether the color is red. Just by looking, by visual observation, you can find out. That is nothing but phenotype or phenotypic. Next, moving on to genotypic. So genotype. So one plant is short, one plant is tall. We can find out by just looking at it. So what are the things that bring about the shortness or the tallness in the plant? It is because of the genes. So that is nothing but the genotype. So genotype. So which gene is responsible for the tallness? Which gene is responsible for the shortness? So we will see how he did it. So tall plants are represented by TT and small t. So here one more term you have to remember is homozygous. Homozygous. So homozygous means this is homozygous wherein both the genes are same. It is homozygous. TT for tall. They are similar. TT for tall and TT, it is for dwarf. Now heterozygous, there is one more term called as heterozygous. So here the alleles are different. That is, the T is one allele and T is another allele. So if it is like this, then it is heterozygous. This allele is for tallness, this allele is for dwarfness. But in spite of that, because T is dominant and this small t is recessive, the plant will be tall. So that is how it is. So this is about homozygous and heterozygous. So he selected here homozygous plants which are monohybrid. That is only one particular trait we are considering, or Mendel considered here. That is, one trait is tall and the other trait is dwarf. So in the F1 generation, what he found out was, we'll do a crossing now by putting the Punnett square. So this is where Punnett square actually comes to help. So have T, then T, here T. We are crossing. Here, because one parent plant is the male, one is male because in one of the parent plant, emasculation was not done. Therefore, both the male and the female reproductive structures were kept intact. Therefore, it is a considered to be a male, and the other one is a female, in which why it is called a female, because here in order to bring about cross pollination, emasculation was done wherein the anthers were removed. So that is what is expressed here. So now we'll make a cross T. So this is the gene or the allele that is going to form T when a cross is made between the tall plant and the dwarf plant. So again here T and T. So here you can see this is homozygous, and here there is no recessive gene. That is recessive gene that is responsible for bringing about dwarfness. It is not there. Therefore, the plant is tall. The resulting progeny, the plant will be tall. So what you need to know here, in this particular experiment, there is no need for us to wait until you sow the seeds and then the pea plant arises and wait whether it is going to be tall or dwarf. Before that only, by putting this particular chart and table, we can understand this because of Mendel. That is why we whoever is studying genetics, they should be thankful to Mendel for this, because he made the research work in genetics very, very easy, or he put a stepping stone to the research in genetics. So here it is heterozygous now, wherein one allele is dominant, that is T, and one allele is recessive, that is small t. But in spite of that, the progeny will be tall. The progeny P plant will be tall. Why? Because the dominant T plant is suppressing the recessive allele here. That is why it was tall. Similarly, here also it will be tall. And moving on to the next one here also it is tall. So sorry, this is small t here, small t, wherein the dominant gene or the dominant allele is suppressing the recessive allele. So therefore, it is though everything is heterozygous here, in spite of that, the plant will be tall because the dominant allele or the dominant gene is suppressing the recessive allele or the gene. So what will be there in the F1 generation? When the seeds are formed and it is sowed, all the plants will be TT, TT, TT and TT. That is, all the plants will be tall. This is a monohybrid cross. So now all the plants are tall. He didn't stop here. Mendel didn't stop here. What he did was, he decided to see if cross-pollination is brought between these, that is the heterozygous progeny, having one of the dominant gene and the other the recessive gene or the allele, what will happen in the F2 generation? That is what he didn't stop his experiment. So what he did again was, so I'll just show it in the next slide or here itself. So F2 generation. Now this is the F1 parent. Now because crossing is done between the F1 plants, it is called as the F1 parent. This will be the F1 parent. Again here also emasculation was done and the crossing was done. Crossing was done. So now he wanted to see what will happen wherein he selected one of the parent of the F1, one of the parent and one of the plant from the F1 generation. So what will happen? Or say for example, TT or small t, anything. So any one of the trait you can use, anything can be used. So we'll see what happens. So here, here we have T, T, again T, T, then T, 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 T. This is a different combination. Say for example, I have used, instead of this TT, a cross is made between these two. That is between the F1 generation itself. So what will happen? Say if this is T, T, and again here we have TT, small t, then it will be TT, T, small capital T, dominant T and recessive T, and dominant T and recessive T, and both here recessive T. So any way you can choose and do. So either you can see for your like for you to understand what might happen, you can again bring a cross between the parent and the what is that F1 generation, or you can make a cross between the F1 generation itself. So anything can be done. But if you bring a cross between the F1 why I showed you is because to make you understand the previous one, if you bring a cross between the F1 generation and the parent, again all the plants will be tall. So what I did again was, instead of doing that, I took the F1 generation and crossed it with the F1 generation itself. So these two are the parent now. I made a cross between these two. When a cross was made again in the F2 generation, so in the F2, now in the F2, what we have tall plant, then we have again tall plant, then again tall plant and dwarf plant. So this is what happened in the F2. So when crossing was done between the F1 parents itself, this is what happened. So here T, it is 1. How many plants were tall? So now you know this one homozygous plant, one plant that is having heterozygous alleles, and the other plant having two plants having heterozygous allele and one plant having recessive alleles, which is homozygous. So one progeny in the F2 generation, when a cross was made between the F1 generation plants, one progeny in the F2 generation will be tall, wherein it will have homozygous dominant alleles or genes, and the other two, they will be also tall, wherein they will have heterozygous alleles, wherein they'll have one dominant gene and the other is the recessive gene. And similarly like that, the other plant, one more plant will be completely homozygous, but here both the genes are recessive, which is supposed to be dwarf. So one dwarf plant. So that is what experiment he proved here. So how many tall plants he got? 3 tall plants. That is in the F2 generation. I'm not talking about the F1 here. So F1 generation, all the plants were tall. What he did was, again to see what will happen in the coming generations, within the F1 generation itself, he brought a cross-pollination, and wherein he found out that by looking at the phenotype, he found out that 3 plants were tall and 3 were dwarf. That is why it is called as 3:1 ratio. It is 3/4 of the plant were tall and 1/4 of the plant were dwarf. And next he looked at the genotypic ratio. So based on the genotypic ratio, the homozygous dominant allele was 1 in number. The heterozygous, here there is a small t, plant having the heterozygous gene, wherein one dominant gene and the other recessive gene, they were 2 in number. And homozygous dwarf plants, wherein they had both the genes or the alleles to be recessive, it was 1 in number. Therefore, he found out that 1/4 of the plant tall with homozygous dominant gene, half of the plant were tall with heterozygous dominant and recessive gene, and 1/4 of the plant again was dwarf with recessive homozygous dwarf gene. So this is what he found out here. So this was about monohybrid cross. So next, moving on, Mendel, he was the first person to say that there are certain factors, that there are some factors which are responsible for the progeny to inherit something from the parent, or to inherit characters or traits from the parent. And he was the first person to say that these factors are called as traits. So earlier, before Mendel itself, earlier thousands of years ago, they knew that one plant will not look like same like the parent plant. There will be some changes, or the progeny will not look the same. It will look the same, but minute changes will be all that. They knew that. But they didn't know that it was because of the genes. And Mendel was the first person to put forward his theory, saying that the characters are inherited by the progeny from the parent because of the transfer of genes from the parent to the progeny. So he was the first person to call them as the genes. And he was the first person to put a mathematical expression, calculate how much in a population of plant, how much plants will look same. So he was the first person to calculate it, wherein he put forward a binomial expression. So what was it binomial expression? We'll study here in detail about it. So he used a formula to calculate. So previous slide I told you, right, a quarter of the plant will be what is that, tall, which having homozygous dominant gene. Half of the plant is also tall, but it has heterozygous gene, wherein one gene or one allele is dominant and the other allele is recessive. And again, a quarter of the plant will have what is that, homozygous dwarf gene. That is wherein both the genes are recessive, and that is why it is homozygous. So how can we say how much quantity or how many number of plants or how many proportion of the plants are tall or dwarf, or how many are having homozygous allele, how many are having heterozygous allele, how many are having dominant genes, how many are having recessive gene? For that he puts a binomial expression. So he was the first person to introduce mathematics into genetics. So what he did was he used a equation called as ax plus by whole square. So please remember your mathematics now. In mathematics, there is an equation called as a plus b whole square, which is equal to a square plus 2ab plus b square. Same thing he altered to his convenience. It is same. Only thing is here instead of only a, ax is there, and instead of only b, by is there. So this, when we expand, it will be ax whole square, which is nothing but a square plus 2ax by, that is, which is nothing but 2ab, and by whole square. So we shall expand this now. So what is a? Here in this equation, a is equal to 1 by 2 and b is also equal to half. He used a constants. A is half, b is also half. Then x is equal to 1 by t. That is the dominant gene for tallness. And same thing, if it is violet color flower with dominant gene, instead of T, we will be writing it as capital W. So it depends on what kind of example we are using. You can alter it based on that. And Y is equal to 1 by small t. That is the recessive gene which is responsible for bringing about dwarfness in the plant. That is small t. So now we'll substitute these in this particular equation. So what we'll get? A, 1 by 2 1 by t whole square plus 2 into what is a? 1 by 2 x 1 by t. Then what is b? Again 1 by 2 and y is 1 by small t plus by square, that is 1 by 2 1 by t whole square. So now expanding this again, 1 by 2 whole square will be 1 by 4 and t again double. Instead of writing 1 by t square, I'll write it as 1 by tt, which is nothing but t square. Because alleles always occur in pairs, that is gene, they have pair of alleles. That is why it is always written like this. It is wrong in this case. Mathematics, you can write it as t square, but in this case always prefer writing it as t, which is also equal to t square. So here 2 will get cancelled. So we'll have 1 by 2 t plus again square 1 by 4 small let 1 by t. So the same thing, it will be 1 by quarter of the tall plus half of the tt plus quarter of t. See, the same thing I showed you. quarter of the plants, 1:2:1. Quarter of the plants will be tall, having homozygous dominant gene. Half of the plant was also tall but with heterozygous genes, wherein one gene was dominant, the other was recessive, and half of the plant, quarter of the plant, again it is homozygous recessive, which is responsible for. So in a population, it is not just Mendel would have selected one or two plants. Among hundreds of plants, it is difficult to understand. So by using this binomial expression, it is easy to solve. So that is what is meant here. So the same thing, it is here. So what he said was, during fertilization, because it is, we can't go against nature. So maybe the progeny, 50% of the progeny will be tall, 50% may be dwarf, 50% if it is tall, they may have homozygous genes, 50% may have heterozygous gene, that is a dominant gene and recessive gene, and again 50% may have heterozygous recessive gene or homozygous recessive genes in them. So all these, to know about these, he used this particular binomial expression to calculate. So wherein he found out that once fertilization takes place, T will be 1/4 of the plant will have homozygous dominant genes for tallness, half of the plant will have homozygous recessive gene, which is also responsible for tallness, and 1/4 of the plant will have recessive gene. So this is what he found out. So Mendel, again what he did, he self-pollinated the F2 plants. So again F2 plants, so I'll show it to you. So F2 plants, what we have now, T, T, T. So again self-pollination. So when he did self-pollination, he found out that if it was a dwarf plant, then all the resulting progenies of the F3 or the F4 generation will be dwarf plants. Because why? Because he did self-pollination to just see what might happen. He did a self-pollination. Again, he found out that in the F3, F4 generation also, all the plants will be dwarf if it is a recessive gene. So that is what he found out. So moving on to two important, after monohybrid cross, he did, again he found out about back cross and test cross. So what is this back cross and test cross? So back cross is when a crossing is done or a cross pollination is brought about by the F1 hybrid with any of the parents. So remember previously when I was doing this, I told you, let me check what will happen if I cross between the F1 hybrid with that of the parent. So what happened? So when a cross was made, so I'll make a cross between the F1 hybrid and one of the parent. F1 hybrid and one of the parent. We'll see what happens. So T, again T, crossing is done between TT, that is F1 parent, T and T. So what might happen here? TT, 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 TT. You see here, again all the plants will be tall. But what is the difference is that 50% of the population of plants will be tall and 50% of the population of the plants will be tall, but 50% will have homozygous gene, which is homozygous dominant genes, and 50% of the plants will have heterozygous genes, whereas when one allele or gene is dominant and the other is recessive. So this is what happens in a back cross. So back cross is the crossing that takes place between F1 hybrid with any of its parent. That is your back crossing. So parent you had already crossed. Now the progeny that you have got, that is the F1 progeny, whatever is there, again you back crossing, you're going behind and crossing it with the parent. That is why it is called as back cross. That is already the parent. You have brought about the cross pollination in the first set of parent plants, and whatever seeds that you sowed, it was the F1 progeny. That F1 progeny, again you will cross it with the parent. You're going back and crossing it with the parent. That is why it is called as the back cross. So I'll make it clear even more here. So this was the parent. So what we did in back cross was, already crossing was done between these two, and here already the progeny was got. Again we are using this progeny, we are going back and crossing it with the first parent. That is why it is called as back cross. So that is back cross. So back cross is a cross when crossing takes place between F1 hybrid with any of its parent. So it is known as back cross. So that is what it's called as back cross. One more we have, that is test cross. So in test cross, when crossing takes place between F1 hybrid with its recessive parent, it is known as test cross. Here the parent will always be recessive. The dominant gene with the recessive parent, dominant homozygous dominant parent with the homozygous recessive parent. That is nothing but test cross. You're testing what might happen. It is the test cross. So this is known as test cross. So we shall study test cross by taking an example. So in order to prove the test cross, Mendel conducted an experiment wherein he crossed between violet and the white flower of the pea plant. So what he did, we will just study. So what is the test cross? As you had seen, test cross, it is nothing but the crossing that takes place between the F1 hybrid and the recessive parent, between the F1 hybrid and the recessive parent. So what experiment he conducted, we'll just see. He took a violet color flower, pea plant having violet flower and a pea plant having white flower, and he brought about a cross with the two. So here this violet flower carries the dominant gene W, and here the dominant gene, the recessive gene W, wherein both are homozygous. So this is the parent plants. So when a cross was done between these two, just put a Punnett square. So small w, that is the recessive allele or gene, and the recessive dominant gene. So when a cross was done, we'll get a heterozygous population now. Heterozygous population wherein the flowers will be violet, violet, violet. This is the F1 progeny. So when a cross was brought in the F1, the resulting progenies, they had heterozygous plants. Heterozygous. So all the population had violet color flowers wherein the genes were heterozygous, wherein one gene was dominant and the other was recessive. So what is according to test cross? According to test cross, it takes place between the F1 hybrid and the recessive parent. So I will choose the recessive parent now. So what Mendel did was he brought a cross again, that is test cross. So you understand test cross means crossing between the F1 hybrid with that of the recessive parent. So Mendel crossed one F1 hybrid with that of the recessive parent. So what happens, we will see. So F1 hybrid and with the recessive parent, he made a cross with the F1 hybrid and the recessive parent. So what happened, again we'll put a Punnett square here. So this is the male W, dominant and recessive. This is the dominant gene, this is the recessive gene, again with the recessive parent, So we have W, that small w, here 2 recessive, W, again 2 recessive. Here therefore we have violet, white, and white. So what is understood here? The resulting progeny will be, that is the F2 population will be violet, white and white. So 50-50. Half is violet, half is white. So that is what we get in test cross. So what will be the result in the F2 generation in a test cross? So in the F1 generation, the result was all the flowers were violet. In the F2 generation, the result was half of the flowers are violet and other half are white. So therefore the unknown flower is heterozygous. The unknown flower is heterozygous. So in this particular condition. So F1 hybrid, or in test cross, F1 hybrid is crossed with the recessive parent. So when the cross is done, in the F2 generation, half of the population will be violet and half of the population will be white. So this was what Mendel explained in the test cross. So in the next coming session, we shall study about Mendel's principles or laws of inheritance, and we shall also study about the non-Mendelian inheritance. So in the Mendel's principles or laws of inheritance, we shall study the laws, like he had conducted various experiments to put forth a theory or two laws. So what are those laws? We'll study there, and we shall also study the non-Mendelian inheritance, wherein non-Mendelian inheritance is nothing but codominance, wherein the genes or the alleles will not get segregated and they will result in a plant having two combinations of genes in one plant itself. So all these we'll study in detail in the coming session. So I hope you have understood the session well. We shall meet in the coming session. Thank you.